I'm pleased to introduce our next speaker, Victor Ujor. He is an assistant professor of food science at UW-Madison. His research focuses on sustainable biomanufacturing, synthetic biology, and microbial metabolism to improve bio-based fuel and chemical production. Please welcome Victor.
All right, I think the other speakers have introduced my talk for me. We're Wisconsin, we make a lot of cheese. With that comes with a lot of whey. What are we going to do with it? In my lab, we're trying to convert whey to value-added chemicals. Essentially, we're interested in butanol and 2,3-butanediol and some other chemicals. For today, I'm going to talk about butanol and 2,3-butanediol. Why are we interested in them? Our interest is purely economical. As a matter of fact, if you look at 2,3-butanediol there, it sells for $2.2 per kilogram. That price was before the price of oil jumped up. It's produced solely from oil today. But if you think about a plant, a mid-sized plant that generates about 12.3 million gallons of whey. Currently, we're making about 85 grams of 2,3-butanediol with whey permeate. So if you add that up, it gives you $7 million a year. So that makes sense for a dairy processing plant. You look at butanol, $1 per kilogram. We're producing 50 grams per liter right now. The same plant we make about $2 million a year. You might wonder, why are we not building plants all over Wisconsin to make this? I have to tell you, this is hot off the press or hot off the lab, unpublished. Over the years, the production of these chemicals from whey has been extremely difficult, and that's what we've been trying to work on to increase production. Most of the time, you find out that the cost of feedstock contributes about 30% of your overall cost. If you use cane sugar, if you use corn sugar, quite expensive. Whey permeate will solve that problem for us. The other reason is that traditionally making these compounds is quite expensive in the sense that the concentration you produce is too low for commercialization. Traditionally, you make about 15 grams per liter with glucose. With whey permeate, that's lactose, you make even less. And that's something we've been working on to come to the point of 85 and 50 grams per liter. So for my work, we work with an organism called Clostridium beijerinckii. It's a solventogenic organism. What does that mean? It makes solvents, but traditionally it makes about 6 to 13 grams per liter, then it stops. It's making butanol, something that's going to kill it. It's an alcohol, like a disinfectant. So we have to find ways to get around that. We get most of our whey permeate from Foremost Farms. Anybody from here today, I would like to say thanks to you guys. You make our work possible. The whey permeate we collect contains about 160 to 300 grams per liter lactose. It contains about 3 to 8% protein. At that sugar concentration, it will kill the organism. So we dilute it down to around 60 grams per liter, add some minerals and yeast extract, and voila, we do our first fermentation. And what we expected was what we got. Low concentration of butanol. Look at glucose, 9.5, whey permeate much lower. We had to ask the question, why? If you look at the organism, the biology of the organism, we say that it's biphasic, without boring you with that pathway. They have the first phase where they make acids. When the acid begins to kill them, they reabsorb it to make neutral solvents, acetone, butanol, and ethanol. In the ratio of 3:6:1. Butanol is the one we're interested in, about 60% of the product. But lactate is not there. So we thought that lactate might be hurting the organism. It's an acid. It can't reabsorb it. It can't use it. Can we get it out of the system? So we used two methods to try to do that. One, a biological means, using an organism called Clostridium tyrobutyrium. It uses lactate as a carbon source. Then we use ion exchange resins to absorb it. Separately, we did this, incubated them for 4 to 8 hours. How much lactate do we have left after that process? Tyrobutyrium, I don't know why this is jumping around. Tyrobutyrium removed about 20%, while our ion exchange resin removed about 80%. It doesn't quite show there, about 80%. So obviously we had to go with the ion exchange resin. But we needed to optimize that. At what concentration can we use it to remove the lactate from the system? We tried 5, 7.5, and 10 grams per 50 mils of whey permeate. You can see clearly there, 7.5 did the best. So we optimized this in the flask. We moved into the bioreactor, repeated it, voila, we went from 6.3 to 12 grams per liter, and that was what we wanted, but not enough for commercialization. We would like to increase that even further. So the next thing we did was to throw some calcium carbonate into the fermentation environment using that treated whey permeate. Why calcium carbonate? Previous studies in our lab have shown that calcium ion enhances the growth of these organisms that make these compounds. One, it enhances sugar utilization. It allows them to be more tolerant to butanol as well. And we went from 12 to 15 grams per liter, which is really exciting. But we would like to increase this much, much further. So we use a process called liquid-liquid extraction. If you think about it, the cells produce something that kills them. They sit in the soup, it kills them. So if you look there in the organic phase, in the aqueous phase, if I can get this to work, over here, is your aqueous phase. The cells produce butanol, it kills them. But if we overlay this fermentation with an organic phase, something that's immiscible with water, hydrophobic enough not to mix with water, but hydrophobic enough to attract butanol, which is organic and hydrophobic. It sits right up there and attracts all that butanol into that particular zone. Here you have low concentration of butanol. The bacteria will be much happier and they'll produce more butanol for you. So we tried that using oleal alcohol, which is a fatty alcohol. It has higher molecular weight than butanol, which means it's easy to separate butanol from it after you've produced it. It's not toxic to the cells in that fermentation environment because it's hydrophobic, it doesn't mix with water. So with that, we tried it. If you look at it right there, it's sitting right on top. The cells are growing very happily below it, and all the butanol, most of it is flowing into that organic phase. If you look at the growth rate here, you can see clearly this is your untreated whey permeate. This is your treated whey permeate without liquid extraction. The cells grew, but if you go over there with liquid extraction, you see a lot more growth, higher cell density. And we tried two approaches, 1 in 10 and 1 in 5 of the oleal alcohol relative to the amount of the whey permeate we used. So did the high cell growth translate into more butanol? Clearly. First of all, look at the treated whey permeate without liquid extraction, about 12 grams per liter, what we got before. Untreated one, around 6 grams per liter. This is the aqueous phase over here. Then you look at the liquid extraction treated fermentation, about 5 grams per liter in the aqueous phase. Where is all the butanol? Over in the organic phase, about 17 point something grams per liter, with 1 in 10, with 1 in 5, around 16 grams per liter. So if you add all of that up, you have about 22 grams per liter and 20 grams per liter, 3.5-fold increase from what we started with, 6.3 to 22. So really excited by that. But that's batch fermentation. Our goal is to add more sugar in form of whey permeate and produce more butanol. And that's what we're doing right now. Still, another thing that is hot off the press. Again, hot off the lab. We haven't published this. It's really new. This is the first time because it's unprecedented. No one's ever produced 50 grams per liter butanol fermentatively. And that's something we achieved very recently by doing fed batch fermentation. Batch means you mix everything together, whey permeate, microorganism, minerals, you grow it, check how much you've produced at the end of the process. Fed batch, you do exactly the same thing. After most of the sugar is used, you add more, and you add more, and you add more. When we did that, we hit 50 grams per liter. We haven't optimized it. We think there's a real potential to increase this to around 70 to 80 grams per liter. That will give us a runway for pure commercialization, so we're really excited about that. The other thing I want to talk about butanol here is that if you look at glucose and butanol, glucose and lactose, whey permeate. The rate of butanol accumulation with glucose is much faster than with lactose. Would like to change that. So what do? We grew the cells in the presence of whey permeate. We looked at all the genes that were upregulated, and we found a sugar transporter, which is a lactose transporter, which means if we overexpress it in this organism, we should bring in lactose much better. Also, there's a gene called an aldehyde reductase in our lab, which we've shown before that when we express it in this organism, we make a lot more butanol. And why is that? If you look at the terminal end of the pathway, butyraldehyde is an aldehyde converted to butanol. We believe that this acts on this. It puts a pull on the whole pathway going this way. And the beauty of combining these two organisms in the same organism is that you bring in more sugar. It doesn't go to making a lot of cells. It goes to making a lot of butanol, which is really what you want. So we use CRISPR to dock two of them together under the control of a strong constitutive promoter and put that in our organism, checked via sequencing. Everything went in exactly where we wanted it to go in. And then we looked at the growth rate. You can see here, this is our wild type. I won't bore you with the whole graphs over there. But if you look at the wild type, it's growing more than our recombinant strain, which is this one here. But our interest is in butanol, and it panned out the way we wanted it to do. It's producing way more butanol. Without liquid extraction, without fed by fermentation, we produce approximately 22 grams per liter. Again, another record, something we're really excited about, that if we subject this strain to liquid extraction, we can produce a lot more butanol than the wild type is producing. But the other beauty of it, if you look at how much lactose is utilized in that whey permeate, untreated whey permeate, this is our wild type, this is our control, 78%. Our recombinant strain, 78%. Then you remove all the lactate in the treated whey permeate, you go from 79 to 100%. All of it is used. That means you can add more sugar. There's capacity to use it. So right now, we're now optimizing this particular organism, this strain rather, using liquid extraction. The other compound is 2,3-butanediol, has huge applications. The organism we work with for this is Enterobacter hormaechei. It uses glucose or lactose, it doesn't care. This organism is extremely promiscuous. Whatever you give it, it will eat it. Makes a lot of 2,3-butanediol, about 42 grams per liter, which is high. But we would like to increase that even further using fed-batch fermentation. But before we could do that, we wanted to look at how we can optimize the process and then scale it up into the bioreactor. And one of the things we did was to remove yeast extract. If we get rid of yeast extract, you're reducing the cost of the production. But most importantly, what do we replace it with? Whey powder. So we want most of our feedstocks to come from the waste from the dairy industry. So when we did that and optimized it and repeated the fermentation using fed batch again, remember batch? 42 grams per liter. Fed batch, feeding extra whey permeate twice, we achieved 65 grams per liter 2,3-butanediol. Then when we fed whey permeate 3 times, we achieved 85 grams per liter 2,3-butanediol. So in all, we've gone from around 6.3 grams per liter butanol to 50 grams per liter butanol. And whey permeate from 42 to 85. So what we're trying to do now is to optimize the fed-batch fermentations. Hopefully we can increase both of them to much higher levels. Well, for butanol, we're looking at 80 grams per liter. For 2,3-butanediol, we hope we can hit around 90 or 95, and that should give us a runway towards commercialization. Along that, we'd like to do some TE and LC as well to try to understand all the things we throw in there, how they affect the cost of the production, and hopefully that will give us a runway as well. I'm interested as well, actually working on setting up a startup with a view to taking this things from the lab, hopefully, to the plant somewhere, and hopefully to the marketplace along the line. I'd like to say a big thanks to Dairy Innovation Hub that make this work possible, and Dairy Business Innovation Alliance out of CDR. We wouldn't do this work without them. We're infinitely very grateful. And of course, a lot of people have worked on this. Some of them are here today. They do all the work. I get to stand here and take all the glory. I'm super grateful to my students and postdocs. Lots of them, I can't mention them all, but I'd like to mention Dr. Ademola Duduyemi and Dr. Omaramike Okereke, who have worked tirelessly on this project. With that, I'd like to say thanks for your time, and I think I have lots of time for questions.
Victor, what's your timeline on getting to 80 grams per liter and doing the TEA for the butanol?
In terms of 80 grams per liter, we expect, hopefully, from the look of things, we're doing the fermentation right now. So it could be in a few days, it could be in a few weeks. So I expect within the next 2 months, we definitely will go way above that 50 grams per liter. Hopefully we'll hit 70 or 80. I would say in the next 2 months. TAA, we're still trying to get extra money in terms of research funding to be able to do a full panel of LCA-TA studies. So I don't know exactly when, but I'm looking to float a startup. Hopefully I can look for venture capital money to do that on the side, not relying on research grants.
What's the budget for a TEA in your mind?
Sorry?
What's the budget for a proper TEA in your mind?
I have to consult my colleague that does that in engineering that I've done some proposals with. I don't want to give a number because it's not my area, so I'd like to talk to those who do it.
So for the recording, that was what's the budget for a TEA, correct? All right, thank you.
Hi, Victor. Very nice talk. Have you considered filing this? This looks very interesting.
I forgot to mention, we already filed a disclosure. WRF is doing some work with us in terms of protecting this. And I mentioned sugar transporter. I didn't tell you what gene that is because we filed a disclosure. We're trying to protect the organism. So yes, we are filing a patent on this to protect some aspects of the work.
So thank you. Thank you. Combining this strain, engineered strain, with your LLE, and have you considered for scaling up a continuous production?
I am not a big fan of continuous fermentation with this particular organism. I'm a fan of fed batch fermentation. We're doing that right now as we speak. So hopefully we'll have results soon. With continuous fermentation, these organisms are very finicky. If you stretch them to a point, they degenerate and so produce only acids. I talked about biphasic growth, acids, and solvents. They will just continue to make acids until it kills them. They don't transition into the solventogenic phase. So we don't want to do the continuous one, but we want to push the fed batch as far as we can to produce as much as possible. So that we're doing right now. You're welcome.
So in your mind, who is your target industry that you want to sell this to? Are we looking at agricultural side? Are we looking at the biofuel side? Are we looking at the
Very good question.
coating side?
A lot of them, 2,3-butanol has probably 100 different applications, but one of the most important ones is used to make synthetic tire. Folks in Akron, Ohio, I've done some work with them when I was at Ohio State. They're really interested in the compound for making synthetic tire. Butanol as well is used to make that, and also, but the big one for butanol is in making paints. It's used in the paint industry a lot, especially automobile paints and lacquer and varnish and all of that. So as I think about floating a startup, I'm working very diligently to identify all these specific customers we could target with that.
Thank you.
You're welcome.
We'll have one more question.
Hi, Victor. Very nice talk. I have a question. In your feed batch process, so how did you manage the accumulations of the inorganic salt and those organic impurities generated from the fermentation that might inhibit your fermentation process, the reactions you want?
I don't think I get your question very well, but we don't have organic salts, so to speak. It's Whey permeate. We have a small amount of salt we add to the environment. It's highly calculated to the tune of how much Whey permeate we have, and a little bit of yeast extract, and we do the fermentation. So we just keep adding the Whey permeate. And it's not Whey, but Whey permeate, so it's already ultra filtered. So we don't have any accumulation of salt with this.